Viruses. Viruses are small, non-living particles that infect living organisms. They are not considered to be living for three reasons. Viruses lack order and are not comprised of cells. They cannot reproduce on their own. They must infect a host cell to reproduce. And viruses cannot metabolize food or process energy. The last mechanism that increases variability in the bacterial gene pool is transduction. Transduction is the process by which DNA is transferred from one prokaryote to another by a virus. Before we go any further, we need to explain what a virus is. Viruses are small, non-living particles that infect living organisms. They are not considered to be living for three reasons. Viruses lack order and are not comprised of cells, cannot reproduce on their own, they must infect a host cell to reproduce, and they cannot metabolize food or process energy. The small dots surrounding this bacterial cell are viruses infecting their host cell. Click for scale comparing sizes of viruses and other organisms and objects. Bacteriophages, also called phages, are viruses that infect bacteria. A protein tailpiece attaches the phage to the host and injects a small amount of DNA or RNA into their host cell. The phage essentially hijacks the gene expression and DNA replication systems. The virus then uses the host cell to make more viruses and kills the cell by causing it to burst open. Phage is from the Greek word meaning to eat. Here is an illustration of a phage. Restriction enzymes are a class of enzymes used by bacteria as weapons against invading phages. They look for specific sequences and pieces of DNA and cut them, thereby destroying the foreign DNA entering the cell. EcoR1 is a restriction enzyme found in E. coli that makes a staggered cut when it reads the sequence GAATTC. The Lytic cycle is a phage reproductive cycle that causes the death of the host cell. The Lytic cycle produces new phages and digests the host's cell wall, thereby releasing the new viruses. Unlike the Lytic cycle, which is detrimental to the host cell, the Lysogenic cycle does not cause the cell to die. In the Lysogenic cycle, the virus's DNA is incorporated into the host's DNA. Then the bacteria cell continues to replicate through binary fission, copying the virus's DNA and its own together. Many viruses are only able to utilize the lytic cycle. Some viruses, called temperate phages, can utilize both the lytic and lysogenic cycles. When a temperate phage switches from the lysogenic cycle to the lytic cycle, it separates its phage DNA from the host DNA and then proceeds through the steps of the lytic cycle as usual. However, sometimes when the virus DNA separates, it takes with it some of the bacteria's DNA. Even though the lytic cycle is a detriment to the individual host cell, sometimes it can be beneficial to the colony of cells. When viruses infect prokaryotic cells, there is a possibility of increasing the genetic variation of the population of prokaryotes. When a part of the prokaryotic DNA is separated with the virus DNA, a beneficial transduction process happens in 6 steps. The steps of transduction. Step 1. A virus infects a bacteria cell. Step 2. Once infected, the viral DNA and associated enzymes destroy the bacterial chromosome. Step 3. The bacterial cell is destroyed and new viruses are released. Step 4. The new viruses, containing a mix of viral DNA and the host DNA, seek out new host cells. Step 5. When they infect other cells, they inject both the viral and bacterial DNA. Step 6. The new host cells take the bacterial DNA from the previous host and inserts it into its own genes.